So students, now we are going to discuss the sexual reproduction in flowering plants. Clear, we know that reproductive unit of the flowering plant is the flower, and we will discuss the various structures which are there in the flower and how they are ultimately leading to the sexual reproduction in plants. Now students, first of all we will discuss the structure of a flower. If we go on for the structure of a flower, we find that this flower is borne on a stalk which is called pedicel. Above the stalk there is a flat structure which is called thalamus, on which the floral appendages are borne. Clear, floral holes are borne. So this flat structure is called thalamus. Clear. Now upon the thalamus there are various floral holes. The first hole you can see here, the green parts, these are individually they are called sepals. The hole is called calyx. Clear, these sepals we know, these sepals have various functions, like it protects the flower in the it is in bud condition. Clear. Next, it being green, it can perform photosynthesis. So these are the functions of the sepals, and this hole is called calyx. Next is the most attractive part of the flower. This is the petals. So next is the most attractive part of the flower. This is the petals. Clear? Individually these are called petals, and the whole is called corolla. The whole is called corolla. Individually they are called petals. They are brightly colored. Clear. Now often they have nectar at their base. They have the various scents. Clear, the aroma glands which secretes this aromatic substances. They have nectars at their base. So they are mainly responsible for protection of the flower, and due to this color, this nectar, this aroma, scent, they helps in attracting the pollinating agents. Clear, like insects, birds. So all these are attracted by this petals. So petals, individually we call them petals. So they are, this whole is called corolla, which is mainly meant for protection and attraction. Clear? They are mainly meant for protection and attraction of the pollinating agents like insects, birds, etc. So they are brightly colored, they have nectar, they often have scent. Clear? And thus it helps in attracting the pollinating agents. So these two holes, that is calyx and corolla, they directly do not take part in the fertilization. So these are called non-reproductive holes. So these two are called non-reproductive holes. Now students will come to the reproductive holes, which are the male is the stamen. The male reproductive hole is the androecium. Individually they are called the stamens. Now if we study the individual structure of a stamen, we'll find that they have a long filament. Clear? And at the tip there is a solen bilobe structure which is called anther. So they have a long slender filament with a bilobe structure called anther at the tip. These anthers, they actually contains the pollen. So when this anther matures, these anthers burst and the pollens comes out. Clear? Which is the male gametophyte. That means it contains the male gametes, and they are released and then they move to the female ones. So stem male hole of the flower is called androecium. Each of the member is called stamen, which have a long filament, and bilobe structure called anther. Anther bears the pollen. Clear? And this pollen are the male gametophyte. These are the structure which bears the male gamete. Mind that pollen is not male gamete, it bears male gamete. Clear? Pollen contains male gamete. Pollen is not the male gamete. It is a male gametophyte, that is a structure which contains the male gametes. So next is we can see here, this is the carpel or pistil. This carpel or pistil represents the female reproductive part, which is called gynoecium. So gynoecium contains numerous individually they are called carpels or pistils. Now if we go on for the structure of a carpel or pistil, we will find that they have a solen base, which is called the ovary. They have an elongated slender part, which is called the style, and above it, it has the receptive part called stigma. Clear? So solen base called ovary, elongated slender part called style, and above it there is a receptive part called stigma. Ovary contains the ovules. Ovules contains the female gametophyte. So mind that ovary is not the female gamete. Ovules are also not the female gamete. Ovule contains a structure which is female gametophyte. Within that there is the female gamete egg. Clear? So egg is the female gamete which is present inside the ovule. But ovule is not the female gamete. Clear? Ovule contains the female gamete egg. Now, so it is there at the base, and at the tip we have a somewhat flattened receptive part. Sometimes it is feathery, sometimes it is sticky, sometimes it is flat. So it is a receptive part where the pollen comes. So this carpel receives the pollen in this stigma. Clear? And the style elevates the stigma so that it can easily receive the pollen which is coming from various agent. Clear? And these pollen, when comes on the stigma, they produces a tube called pollen tube which fertilizes, which moves inside the ovule, and finally the male gametes fertilizes the egg. Now there is a step before fertilization, because this step is called pollination. What is pollination? Pollination is the process by which the pollens from the male, this reproductive part, that is anther, is brought into the female reproductive part, that is stamen. So pollination is the process where the pollen from the male reproductive part is transferred to the female reproductive part, that is the carpel. We know that stigma receives it. Now this may happen that in both the anther and stigma are of the same flower. So it is transferred from the anther to the stigma of the same flower. Then we call it self-pollination. Clear? Then we call it self-pollination. So if the transfer of the pollen from the anther to stigma occurs in the same flower, then we call it self-pollination. And for self-pollination, the flower must be bisexual, obviously. Clear? They have to be bisexual, and generally the pollination occurs in bud condition or least exposure is there. So this type of pollination we find in certain flowers like camelina. Now in other cases, the pollens from the anther is carried to a stigma of the different flower. Pollen from the anther is carried to the stigma of the different flower, and this is called cross-pollination. So this is called cross-pollination, where two different flowers, maybe two different plants, it may be two different flowers of two different plants may be involved. Clear? So this is cross-pollination. And now if we go on for the fertilization in angiosperm, we'll find that angiosperm has these flowering plants, basically the flowering plants, angiosperms. They have a unique process of fertilization where there are two unions. Clear? In angiosperm we find two unions, because angiosperms are having two flowers and there are two unions. In one case, the union is between the egg and the sperm. We call it, and they form the cell called zygote. Clear? Which ultimately develops to embryo, and embryo develops to new plant. In the other case, they have the fertilization between the sperm and the two haploid cells present in the female gametophyte, which is called a central cell with two haploid nuclei. Clear? So that formation, that helps in formation of a triploid cell called endosperm, which divides to form the endosperm cells, and endosperm provide the food to the growing embryo. So in angiosperm or flowering plants, we can see double fertilization. That means one sperm fuses with the egg and forms a diploid cell called zygote. This zygote ultimately develops to embryo. Embryo finally develops to new plant. In other case, the sperm fuses with the central cell containing two haploid nuclei, and that ultimately, so here sperm one haploid nuclei, here two haploid nuclei of the central cell, they fuses to form a triploid 3N cell, which is called endosperm, primary endosperm cell, which divides and redivides to form endosperm and provides food to the growing embryo. Now after fertilization, these ovule, clear, which contains actually which is the structure which actually contains the egg, clear, these ovule now develops to seed. So after fertilization, these ovules which contains the egg where the fertilization has occurred, it develops to seed, and the ovary, this is the outer covering, clear, which actually sustains the ovules, they develops to fruit. Clear? The ovary develops to fruit and the ovule develops to seed. Clear? Now, if we go on for the structure of a seed, what we'll find? We'll find that it has a outer covering which is often hard. You can think of gram seeds. They are often hard, and that is called testa. Inside the testa, there is another fine structure called tegmen. And if we go on for the surface of the seed, we'll find that there is scar, clear, scar-like structure, which is called the hilum. Now there is a pore through which oxygen, water enters during the germination. This is called micropyle. So this is the outer surface of the seed. And now if we cut it, what we are going to suppose? So suppose this is a pea seed. So this is the outer surface having testa, outer covering. There is a scar called hilum. There is a pore called micropyle. This is the outer surface. Inside it you will find two fleshy, think of pea, you will find two fleshy which we eat. These are the two fleshy cotyledons. And there is the initial root. This is the part which will grow to root is coming out. This is called radicle. And there is another part which will grow to shoot, will coming out. This is called plumule. So this is the embryo with radicle and plumule, clear. And from this cotyledonary axis, there appears two cotyledons which actually stores food for the growing embryo. So as the embryo will grow, these will store the food, and until and unless it germinates, the seed germinates and they starts photosynthesis, they will provide the food for the growth of the embryo. So you can see here, so this is the gram seed, clear. This is the micropyle, this is the scar, and this is the outer covering, hard covering called testa. Inside it there are two cotyledons, and this is the embryo. So this type of seed where there is cotyledon, clear, the food is stored in the fleshy cotyledon. We call it non-albuminous or non-endosperma seed, because here the endosperm which has been formed during fertilization is used up before it forms the mature seed. Clear? So this type of seeds are called non-endosperma seed, which we find mostly in the pea, bean, these dicot plants. But if we go on for the if we go on for the seed of the rice, wheat, etc., this monocot plants, we will find that here they have the, think of maize, there is the embryo and there is that endosperm, clear, which is still retained in the mature seed. Clear? So this type of seeds are called endospermous or albuminous seed. So here it is the embryo and this is the endosperm. Clear? So here in the mature seed also we find endosperm. So these are called endospermous or albuminous seed, which we find in monocots like rice, wheat, maize, all the grains generally which we eat. Actually there is a storage of carbohydrate in endosperm. Clear? And these seeds, dicot seeds, here the endosperms are generally used up, clear, before the seed matures, and so the food for the growing embryo is kept stored in the cotyledon. So these seeds are called non-endospermous or non-albuminous seed. So student, till today, till this, hope you have understood. Okay, thank you.